In today's class, we are going to run an experiment on transportation across a cell membrane. And to do so, we were able to make a model of a cell, and we looked at the movement of particles across that membrane. Now, to do this, we used a product that is very similar in properties to a cell membrane. This tubing is called dialysis tubing, and it has properties that allow the movement of some molecules across the membrane. And in our cell, we decided that we were going to put some solution with different molecules. So our solution that we entered in was a solution that included some water. It included some glucose, which is a simple sugar that gives us quick energy. And we also added starch, which is a complex carbohydrate that gives us longer term energy. It's a bigger molecule, so it takes longer to break down and release energy. So we had this solution and we pipetted it into our cell. We tied off the ends and we had our model here. Now, in our extracellular fluid, outside of the cell, we placed our cell in a solution of iodine. So a little combination of water mixed with some iodine that we added to this solution. Now, the interesting thing is that iodine works as an indicator in the presence of starch. So if I take some of this iodine solution and I put it into the graduated cylinder, you can see that it has this golden red color. And if it comes into contact with starch, it changes color from red to black. So similar here, with the absence of starch, iodine stays golden red. In the presence, it will turn this dark blue-black color. Our cell was taken and it was placed in the solution for 30 minutes. And being that we were talking about the cell membrane, we made predictions on which particles would move across the membrane. So in our first part of the lab experiment, we determined whether there was a movement of water in or out of the membrane. And then we used our results from the starch and iodine to determine whether the starch molecule was able to move across the membrane. And for the next part of the lab, which you'll see in future videos, we also wanted to determine what happened with the glucose. So we tested that glucose later on using a different indicator, and you'll see that play out. It was called Benedict's solution.